is that the enzyme will go up the DNA reading 3 at a time, and it'll keep adding new Rs, new amino acids until it hits that. Once it runs into that, it just releases the protein and it gets transported. Now, I'm going to go into a lot more detail next time, but the main thing to get today is I'm not good with faces, so I don't know. Is Phil here today? Thank you so much for your email. It made an enormous difference, as you might have been able to tell by my second attempt on YouTube that I mentioned to you. I'm planning on turning my handouts into a book, and the book will contain a CD with all of the audio recordings and all the images on it and all the handouts. So I'm trying to make the audio, trying to get the audio up onto YouTube with all the images so that you can see it and make some sense of it. Okay, it's 11 o'clock. I promised at the beginning of this class that I would try to take breaks, so let's take a break now. Are there any questions?
Yeah, you asked for a subject, in your email. What I had in mind is if you start with, we were just discussing together here about the original, I saw the cell started the world.
I can tell you the theories. I can tell you the hypotheses, excuse me.
Yeah, would you?
Yeah. We can do that for the last class.
Yeah.
That would be kind of fun. How did life actually begin? How do we think the chemistry came together to form life on this planet?
Yeah.
And it's good that you're telling me that now, because it's going to take me 2 weeks to get that information together into a handout. There's a lot of stuff there. But we've already talked about things like cosmology and astronomy and time and radioactivity and dating things. And you have to, it would be a good idea for that last class if you just skimmed through all of the handouts 1 through 7 before you come to that class, because you will need the awareness, and you'll need a lot of that stuff in your head to begin to follow that last class, because it's going to be, it will be challenging. Now, good. We've had enough time for the experiment to do the next step. Okay, so here I have mushed banana with a little salt water, and I've added some soap. What does soap do to the cells in the banana? Okay. What is a cell? I'm exaggerating. This is a cell with the nucleus and the cytoplasm, and this is the membrane. What is the membrane made up of? If we were to take a segment of this and make it much bigger and put it in a straight line so it's easier to see, you would have segments of a fatty acid which like water, so they don't mind being on the edge, because the inside of the cell is mostly water, some salts, and all those proteins. But it has a segment attached to it which doesn't like water at all. Same on the other side. There's a piece of a molecule that likes water connected to a piece that doesn't. And so the pieces that don't like to be together because they squeeze all the water out by repelling it. And so you end up with what's called a lipid bilayer. And I will have to, actually, you know what I'm going to do? Pardon me. That'll remind me to put it in the handout. But the entire thing is surrounded, the entire cell is surrounded by this kind of a layer. However, there are proteins which cross this barrier completely. What do you think those proteins do? Well, I'll give you a hint. Some of them are hollow. Does that bring anything to mind? These are transport proteins. These are things which make whatever's inside go out, or whatever's outside come in. They all of the proteins that are in the membrane have, let's call this in and this out, inside and outside of the cell. All proteins that cross across the membrane, or just sit on the top, or just sit on the inside, will have things that are recognizable. And so, for instance, a hormone may come along and glom onto this, and that functions as an on-off switch. So you may have something that comes up and says, do your work, go. And so it will change the shape of the molecules in this protein such that they'll be active. Or it may attach, and it may actually cover up the opening, or make a change in the conformation of the protein so that it stops functioning. And these are antigens, which is one of the words that's not in your glossary. But you've heard of antibodies. Well, antibodies are proteins which fight against bacteria and viruses and all the baddies. They also take care of cells that are dying and remove them before they turn into a cancer, for instance. But it's also where things like the viruses attach. So if you get a rhinovirus, a cold, the reason it's in your nose is because the nose cells in the mucus lining have specific antigens on their surface that the rhinovirus attaches to. That's why rhinovirus doesn't usually give you indigestion or warts or some other thing. You could inject rhinovirus under your skin and absolutely nothing would happen except that your body would get rid of it. You wouldn't get sick at all because it's in the wrong place. Is everybody clear on this one?
Can you determine beforehand which areas would be
visible?
Susceptible? Yeah.
Visible? Well, one of the things that you can do is you can purify this protein, and I worked in a research lab where we did that. There are a number of things you can do. It's really kind of fun. For instance, you do something like the experiment we're doing today, but you use different proteins so that what you would do is you would cut up and eat up all of the cell membranes and you get the proteins out of them instead of the DNA. And you can put it into a column where there are little beads that have different size holes. So small things would get stuck in the holes and big things would go down first. So you put it through that and you drip it out of the bottom and you drip it into tubes, 10 cc's per tube or something like that. And tube 1 will have really big proteins and tube 101 will have really small proteins because they would have gotten caught in all those little holes. So you can purify just this protein in the lab, and then what you can do is you can inject it into a rabbit. And the rabbit will tell you by its antibodies all of the things that are visible on that protein. And that's one of the difficulties that we have with with influenza, is that influenza mutates at such a high rate that that we can't keep up with it.